those things where you don't want to be looking at your phone, stuff like that, you know? So anyway, with that, I'm going to go ahead and get started. This is my freshman writing seminar, SSI 165A, and our nickname is Never Really Alone, Symbiosis and Parasitism Within and Around Us. And it is my distinct pleasure, my class, to introduce you to Professor Margaret McFall-Ngai, who is a leading expert in symbiosis. She has an amazing CV. I keep forgetting that she was heavily involved with the American Society for Microbiology. She's a member of the National Academy, a Guggenheim Fellow, just an amazing natural force in the study of symbiosis, and who better to have speak with us today. As usual, what I have done, I should say, and that we overlapped at UCLA. She was a graduate student when I was an undergrad, and no, I never had her as a TA. Because at that time, she was more of a field biologist. You did work in the Philippines, is that right, Margaret?
That's right.
That's right. And then from there, she began working on a number of different systems until she settled on that wonderful, and Alice, this is for you, symbiosis between Vibrio fischeri and Euprymna scalopes, the Hawaiian bobtail squid. And she is a leading authority. In fact, I would call her the authority on that symbiotic association. But further than that, she is even more of a microbial supremacist than I am. And that's saying something. And so she really has done a number of wonderful position papers, of which you have read one, on this entire subject. And so today I thought it would be wonderful if she had a look at the questions that you have given her, and she will respond to them. Does that sound about right, Margaret?
That sounds great.
I apologize again for a lack of picture.
Not a problem. Okay, well, so should I just start talking?
Absolutely.
Okay. Well, first of all, I just want to say thank you for giving me this opportunity, Mark, and all the students in the class. This is just really fun. And so the other thing I wanted to say was that you guys are amazing. These were great questions, and I had a great time sitting and thinking about how we would talk about them and so on and so forth. The first thing I wanted to say, though, was that I think it's really important for you to recognize that I actually did not write that paper, Animals in a Bacterial World. So what happened was I was fortunate to have this Guggenheim Fellowship to think about this sort of thing. And as part of that, what I did was I got together 25 people from around the world to come to the National Evolutionary Synthesis Center at Duke University and to write a paper. So I've never been to a meeting where all you did was write a paper. Nobody talked about their work. What we did was we divided into groups of 5 that in each group they were specialists in a given area. And they were told that they had 4 days to come out with 700 words and a figure. And so each day they would go their separate ways, and we'd come back to, excuse me, at the end of each day, and we would talk about what we had done that day. So it was phenomenal. And in fact, I was very much in favor of producing an article in which the authors were alphabetical. And I fought very, very hard for that. And I was completely and utterly overruled. And basically, what happened was, at the end of the day, it was myself and Mike Hadfield who took all 5 sets of writing and put it into one voice and sent it in to Science Magazine. And actually, we sent it into Science Magazine. And because it's very controversial, we got huge pushback from Science Magazine. And you are talking to a person, probably the only one that you'll ever know in all your science career. My authors pulled an accepted paper from Science Magazine because Science wanted to keep rewriting it in the way they wanted it to appear, and the authors were not happy with what Science was doing. So we pulled it and asked Dave Curl to put it in PNAS, and that's the article that you saw. So I just wanted to get that out there so that you knew how that paper came about. But the lesson there is that there are politics in science. And there is not a small number of people who would like not to have this sort of thing as visible as it is. And in fact, it's a very exciting time in biology, and my training is in animal physiology. I'm not a microbiologist, and yet it's very clear to me that microbes are at the base of everything. And biology has to change a lot if they're gonna go there. So what I'd like to do now is I'd like to look at some of these questions. And since Mark mentioned at the very end of his introduction the squid, I got one question on the squid from Alice. And so I'd like to address that question first, just to get that out of the way. It's, of course, my favorite topic. So Alice, do you want to read your question?
Alice's mouth is full. Do you want to read it, Alice, or not?
Oh, I can read it then.
Okay.
You want me to read it? Okay. So Alice asked, I've read that the bobtail squids shed up to 95% of their bioluminescent bacteria Vibrio fischeri every night. This seems counterintuitive for the squid to be recolonized each day. What benefits does this have for the squid or the bacteria? So the bacteria are the remaining 5% that are left behind after the animal vents 95% each day. That remaining 5% grows back up to fill the light organ each day. And there are various theories. One is that why have all those bacteria in there and feed them all day if you're going to be in the sand not using their luminescence? That's one possibility. Bacteria do continue to grow, and so very, very slowly, but they continue to grow in the light organ, so there's got to be some mechanism for the control of symbiont member. So one possibility is that you leak them slowly all the time, or when you don't need them at dawn when you're burying in the sand, you can get rid of most of them and then grow them back up. The other thing that I've heard is that maybe it protects against cheaters. In other words, the growth of dark mutants in the light organ. I actually don't believe that one.
That's too bad because I kind of do.
Well, so here's the deal. The animal is exceptionally good at picking out cheaters. So if you colonize a baby with a delta lux mutant, that is a mutant that's defective in light production, the animal takes it in, and after 24 hours, it starts to get rid of it. And it says, there's something wrong, this microbe is not doing its job, it's out of here. Okay, so how does the animal know that the bacteria are making light? Well, the light organ has all of the biochemical components of the visual transduction cascade in the eye. And so it has rhodopsin, the visual pigment that allows you to see in the eye. So the light organ has rhodopsin, and we think that that's how the animal perceives bacterial light. So it itself, from the very beginning, is exceptional at getting rid of cheaters. So I don't think that the everyday thing, the everyday venting, has to do with cheaters. I think it has to do with the control of symbiont member. But that said, nobody knows for sure.
And the little squid are incredibly cute. I've macerated a few of them myself.
They are. They're incredibly cute. Okay, so how about Gabe Whitson?
Gabe is front and center. Say hello, Gabe. Hello.
Hi, Gabe. So your question, could you read your question?
I actually don't have my question in front of
Oh,
me.
you don't? Okay, I'll read it for you. So you said, do you have any ideas of what evolution would look like without our microbial companions? Would it be possible to evolve anywhere close to where we are now without them? It would be interesting to study the evolutionary change between two groups of animals, one with microbes and one without. Well, this is a really interesting question. And I started thinking about it and spent quite a bit of time musing on what evolution, would it be possible to evolve close to where we are now without microbes? And I actually think the answer is no. And so it's almost like Well, so when we look for life on other planets, like Mars and whatnot, we always look for small things that, like bacteria, single-celled organisms, or things like, I don't know if any of you have seen the Andromeda strain, but life forms that are what would not multicellular, not with that sort of thing. So at some level, I don't want to say that microbes are not complex, because in fact, in some ways, they're much more complex than eukaryotic cells, because the entire metabolic scope that's possible for life forms on Earth seem to be represented in the bacteria. But it's that, imagine, so multicellular organisms, I think, like animals and plants, have a certain type of complexity. So imagine if there were cars on the road all of a sudden. They just appeared out of nowhere. I started thinking about that. It seems like you first have to invent the wheel, and then you have to invent the various parts and the various functions, and then it sort of evolves out of that. So I think in very much the same way. I just don't think that animals could just appear the way they are. Now, it might have happened differently, and probably does on other planets, but I think that that what we have in the multicellular organisms on Earth could not have happened just by spontaneous generation. We would have forms like we have. Evolutionary changes in two groups of animals, one with microbes and one without. There's some evidence to suggest that C. elegans. Do you guys know what C. elegans is?
Yeah.
Yeah, it's a nematode worm, and C. elegans eats bacteria. And there's some evidence that while you can sometimes find microbes in the gut of C. elegans, that they don't normally have a microbiota. Now, it's actually kind of unusual. Most animals have, and it comes from the fact that all animal body plans arose in the oceans. And the oceans are an environment of 10 to the 6th bacteria per mil, which was probably at least that in the ancient ocean. So 1 million bacterial cells per mil. So there's a pretty good biotic force there, and 10 times as many viruses. So there's a lot of microbial life interacting with animals as they evolved in the oceans. So it's very natural to form alliances because microbes can do things animals can't do. So it's, why not form alliances? It's interesting, when animals made the water to land transition, came onto land, they relegated all of their mucous membranes to the inside. And they do have, oftentimes they have microbes on their skin or so on. But they're a lot less interactive with the microbial world and the terrestrial world. And so C. elegans, it would seem, has arrived at some kind of strategy of having a cuticle on the outside and not having very many microbes associated with it. And it's kind of interesting because if you look at C. elegans and you look at its genome, it's lost a whole bunch of pathways that are associated with interacting with microbes. So that's what might happen. It would be to not evolve if you're a group of organisms and you have very little interaction with the microbial world, and that's your strategy. That would be, in my mind, considered a derived condition. In other words, lost. They've lost the ability, and that's what C. elegans, the studies of the C. elegans genome would suggest. And also, the other group of organisms to evolve with microbes, closely related to C. elegans, in other words, in the same superphylum, the Ecdysozoa, are the insects. Now, insects have lots and lots and lots of microbes. And so, for instance, 11% of all insects have intracellular symbionts in certain cells of inside their body cavity. And those 11% have these microbes so that they can live in environments that are nutrient poor. So, for example, aphids have them. And the presence of the bacteria allows the aphids to live on plant sap, which is very low in nutrients. So the bacteria provide them with certain amino acids and various other nutrients that the plant sap doesn't have. And so 11% of insects have this, insect species have this particular behavior. And as you guys have read, it's also true that in some places up to 60% of insects have, what do you call them, Wolbachia. And Wolbachia can sweep through populations. So insects, unlike C. elegans, can seem to have a lot.
You know, Margaret, that's very interesting to me. I didn't know that about C. elegans. And the thing that's funny about it to me is that you can do RNAi, so you can genetically engineer bacteria, feed it to the roundworm, and it picks up the DNA. So it almost makes me wonder if there's a whole bunch of passports in order to get genetic information partitioned between organisms, and C. elegans has lost theirs.
That's a very interesting idea. You know, it's so amazing to me. The other thing that's really amazing is because C. elegans is a phenomenal genetic model. And so what people are trying to do is they're trying to crowbar C. elegans into a model for beneficial microbes. And so they'll go out and they'll look in the soil and they'll find C. elegans, they'll grind it up, and they'll find a bunch of microbes. Well, they don't ask the important questions. Are the bacteria dead? Are they, you know, they so badly want to look for their keys under the lamppost that they try to crowbar it into having microbes. But I think what I think we ought to do is I think we ought to embrace the fact that there are likely certain animals that have a strategy not to have microbes associated with them. And we should study what they've lost. And study, you know, we can learn a lot by studying the exceptions to the rule, not just the rule.
Crazy.
Okay, so should we go to the next question?
Sure.
Okay, so I sort of want to stay on the question of evolution. So Amaya Rodriguez, is it Amaya or Amaya? Amaya?
She's right here. It's
Okay.
hers, right, Amaya? Yeah.
Do you have your question? Do you want me to read it or do you want to read it?
You can read it.
She wants me to read it? Did
She
say?
does. She does, Margaret.
Okay. So Amaya asks, how do you think a bacterium would have come to naturally evolve into being necessary for a species to survive? Which came first, the insect or the bacteria? How could they come so far as to evolve at this level? In other words, you see these really complex symbioses. How could they have gotten started? Well, there have actually been some really interesting experiments with this. There was a professor at the University of Tennessee named Quan Jiang, and Dr. Jiang did an experiment with amoeba. And there were bacteria associated with the amoeba that were actually pathogens of the amoeba. And what he did was he grew the amoeba and the bacteria together, and over time, the bacteria became less and less pathogenic of the amoeba, and eventually the amoeba could not survive without the bacteria inside it. And so he was able to evolve the symbiosis in the laboratory. And so that's an example. Now, symbiotic associations go everywhere. So the word symbiosis is a word that applies to all types. All it means, by the strict definition that DeBerry gave it in 1879, is it's organisms living together. And symbiosis can be anywhere from mutualism, where both partners benefit, all the way to parasitism along this spectrum, where one partner benefits and one is harmed. And I think that a symbiosis can arise anywhere along that spectrum and then be selected for over time. And so one of the other ways that this could happen is, let's say in the luminescent fish symbioses, they're almost always diverticula off the gut. The light organ symbioses in fishes will be little pockets off the gut. And they're often, all the luminous bacteria are have chitinases. And all of the animals that have luminous bacterial symbioses are planktivores. In other words, they eat plankton with chitin. And so probably the ability to break down chitin drove the selection pressure on the association between the microbe and the host. So the microbe helped the animal digest something. So then you start amplifying the numbers. Then you make a little diverticulum off your gut. And then another thing that could happen is, if you make that an obligate symbiosis that you pick up right as you hatch, the way the squid does, one thing that might happen is you could slowly lose expensive metabolic pathways that the symbiont could do for you. So, for instance, tryptophan metabolism, or something that is tryptophan synthesis, or some other metabolically expensive synthetic pathway that the bacteria are very good at. You just say, wait a minute, I don't need to do that anymore. I'll let my bacteria do that for me. And then slowly, you become more and more dependent on your symbiont. Does that help?
Yeah, thank you.
Okay, good. Okay, well, sort of staying in the same vein, I wanted to now, let's see. There are a bunch of you who asked about antibiotics and extinction of bacteria and so on. And so I wanted to start with Katie Maletta. Katie Maletta?
Say yes.
Yeah, hi.
Hi, Katie. So Katie had two questions. One was, referring to the gut microbes, there was a statement about dietary influences. Americans needing a high-fat and high-protein diet versus others who consume carbohydrates. If we keep feeding the wrong microbes with the right microbes, will the good microbes eventually become extinct? So just think about it. Keep that in the back of your mind. Might the good bacteria become extinct? So I'd like to now mention Emma Casey's question. Emma, are you there?
Well, Emma apparently couldn't find this room.
Oh, she couldn't find this room. Okay.
But
So Emma also asked about extinction. So in very much the same way. And then Matt Sherman. Matt, are you there?
Yeah.
Yeah, so Matt asks, are there, let's see, as stated in the article, Microscopic Menagerie by Susan Milius, the specific bacteria inside insects that can dictate whether the species lives or dies, like Wolbachia in bedbugs and Wigglesworthia in the tsetse fly. Are there any bacteria, like Wolbachia or Wigglesworthia, in the human body that are essential for survival? If so, could the use of antibiotics in today's society affect said bacteria? Well, so I wanted, let's see, I think there's one more, and that's Darby Bosco.
Yeah.
Yeah, and you also mentioned extinction. And so I thought I'd just spend a little bit of time talking about this issue. So it turns out that, have you guys heard of Martin Glaser?
They have not heard of Martin Blazer's book about Helicobacter pylori, and
Right.
that in some cases it's responsible for peptic ulcers, but it also can be thought of as a mutualist in some circumstances.
Yeah. So Martin Blazer is a professor at NYU, and he's the head of infectious disease there. He's a physician, and he's actually a very interesting guy. He's also the chief physician to the UN, and he wrote a book that just came out this past April called Missing Microbes, and I would highly, highly recommend it. And what Martin Blazer talks about is he talks about the fact that antibiotics are the overuse of antibiotics is resulting in us losing essential microbes. And we are becoming a country that is full of allergies and asthma and whatnot that actually didn't exist 50 years ago. These conditions were rare. So I can tell you that when I was a kid, there was nobody in my class who had asthma. There was nobody in my class who had allergies. And, if my mother couldn't make a peanut butter sandwich for the kids, she would, that was the easiest thing to make. And I guess nowadays you can't even bring peanut butter to school. So the point is that we're becoming prone to autoimmune disease, allergies, and so on, and asthma. And it's thought that this is due to an antibiotic-induced imbalance in the microbiota, or other reasons for our loss of the microbiota. So what Martin Blazer points out is he points out that in the turn of the 20th century, in the early 1900s, in the United States, there was about 90% carriage of the microorganism called Helicobacter pylori in the stomach. And now about, 15 years ago, Mark, or 20 years ago, it was found that Helicobacter pylori is the microbe that causes ulcer and can cause gastric cancer. But it only does so at any high levels in individuals who are...
Oh, we froze! Margaret! Oh, no. Are you back?
Yeah, what was that?
We had a freeze. You're back.
Oh, we're back. Okay.
Because it was a great stoppage. What was the last thing she said before it froze? It was when Helicobacter only causes disease when, and then your face froze. It was great.
Oh, okay. when an individual is post-reproductive. And so what that means is that, it happens to people after they're 50. And so what that means is there can be no evolutionary selection pressure against the carriage for Helicobacter.
How did I miss that? Margaret, how did I miss that? That's fabulous.
And so what has been found is it's been found that in various experiments with laboratory animals, that the carriage of Helicobacter confers resistance to getting asthma. And there are a whole bunch of different, reasons why it seems to have been a coevolved microbe. One thing you can show, for instance, is you can trace human populations and the diversification of human populations around the world more accurately by studying the Helicobacter genomes, the genomes of the Helicobacter that they carry, more than any of their own genetic characters. And it's Martin Blazer who's shown this. And so Marty Blazer has this wild idea so I should say that now only about 10% of the Americans carry Helicobacter pylori as children. Only about 10%. And in the non-industrialized nations, there's still 90% carriage or so. And so what's really interesting about that is there is no gastric cancer or ulcer in the non-industrialized nations. So So we've got some really interesting characters here in the industrialized nation. So Marty has this really wild idea that we should give all babies Helicobacter. And then if later on in life they get gastric cancer, then you can cure it. So this is pretty wild stuff. But the idea is that Marty is worried that we're driving some of our microbes to extinction, and some of the ones with which we evolved and that keep us healthy. The other thing is that one of you guys mentioned that what happens when an animal goes extinct? Is it a big deal if all of its coevolved microbiota go extinct? Well, that can have pretty good repercussions, because remember that a lot of the symbioses are horizontally transmitted. And so, is Emma Brammer there? Brammer. Sorry, Emma. So Emma asked, do you think that there could be a possibility that all species have at least one bacterial species required in order to reproduce? So that is actually a very tricky question. And the reason it's a tricky question is because symbioses are divided into two different types. They're either horizontally transmitted or vertically transmitted. And most often, the definition goes as follows. A horizontally transmitted symbiosis is one that is picked up anew each generation from the environment. And so in the squid that I study, it picks up its symbiont right after it hatches. It's thought, although this is becoming more controversial, it's thought that when we're born, we're not born with our symbionts. And that we acquire most of them from our mother and so on. And we are thought that most of our microbes are horizontally transmitted. Vertical transmission is when the animal, the female, puts the bacteria in or on the egg. And the bacteria participate in the events of embryogenesis directly. And so in vertical transmission, there are lots of invertebrates, like many insects do vertical transmission. They put their bacteria in or on the egg. So in horizontal transmission, though, even though the bacteria don't take part directly in the events of embryogenesis, when the embryo is forming, it has to form the receptors and all the various things necessary that when the animal hatches or is born, it interacts with the right microbes in the environment. It can recognize right away the right bacteria. So that those characters have to have been developed embryonically. And so, yeah. The other thing is that is this idea of, do you require your symbiont? So in the lab, the squid doesn't require the symbiont because there aren't any predators around. And physiologically, without its symbiont, it seems just fine. And light production, it doesn't seem to supply the animal with a nutrient, but rather with light production. And so in that way, it's a character that doesn't seem to affect the health of the animal if they're not there. But boy, are they obligate in the ecological context. When the animal's out in the field, we have never, in 25 years of collecting these animals, ever found an animal without its symbiont, which suggests it's really important. Okay? That's all about...
Margaret, can I ask a question real quick?
You can.
Yeah, it's Mark. So I'm interested in these animals that undergo cryptobiosis, tardigrades being one, Artemia another, that kind of thing.
Right.
I wonder if they carry their if they, number one, if they have a specific coevolved microbiota, but if they do, then it has to be carried along with them in their estivating state. Do you think maybe they're like Caenorhabditis and they don't have a microbiota?
Well, it's my understanding that tardigrades do, and they do have a microbiota. Artemia, I'm not sure about. One of the things I find really interesting, though, is that people who study anhydrobiosis, like Steve Hand at Colorado, don't really think about the microbes that might be with the host. And I'm actually did a postdoc with George Somro, and Steve Hand did a postdoc with George Somro, and we all of his students just went to his retirement party. And one of the things we did, I was asked to talk about microbes, and I was the last speaker. And at the end, I said, all you guys, lots of the things you're talking about are just screaming that there are microbes involved. And I think that some of these things, for instance, the proteins that allow for anhydrobiosis and the glycopeptides that allow for freezing resistance, is it possible that some things like peptidoglycan monomer can be exported? The structures of those molecules look very similar. And so I wouldn't be at all surprised if microbes were involved in some of these processes, just that biology has been so siloed that the microbiologists don't talk to the animal biologists much, and vice versa.
I guess I was thinking about it because the slime mold dictyostelium carries along the bacteria it prefers to eat, which goes faster than it does. So they're called farmers, and I think of them more as packing a lunch.
Yes, yes.
But I mean, that makes me think of all this like nesting box view of things, which is just fascinating to me.
Oh, yeah.
You have to tell me later about microbiota and tardigrades, because I've been looking at that. So let me know later about that if you could.
Very cool. Okay, well, let's see. So Ellen Hahn, are you there?
Hi, yeah.
Hi, Ellen. So you and Tori Butcher asked about issues about diet and so on, and whether or not microbes are stable, and what the effect is of having different genetic background. So the first thing is that what the Human Microbiome Project has found is that each individual has their own microbiota. And so they're like fingerprints. Now, that is not to say that there isn't a genetic proclivity to associating with certain microbes. And so it turns out that my microbial guild structure is more like, so I'm Irish American, and my microbial guild structure is very much more like the people in Ireland than it is like the people I work with here in Madison, who are mainly German. It's interesting that genetic proclivity is there. It just is there. But you establish your own microbiota anew each generation, and after the ages of 2 to 4, it is extremely stable. And that's why it's so, if you take probiotics, you can't just take them and hope that the bacteria establish themselves. You might be able to do that with an infant before the age of 4, but you can't do it with an adult. And that's because the ecological sites for the bacteria are already occupied. So you can displace them, or you can put bacteria in there that will grow and change the environment transiently, but it's very hard because your microbiota is extremely stable. Now, what happens if you go from being a carnivore to a vegan? The microbiota responds extremely quickly and changes extremely quickly, not in its composition, but rather in its ratios and proportions. So the same guys are in there, it's just there are more of one type than there were before. So you get between the ages of 2 and 4, it gets very stable. So Tori asked about if people have had a food that they've enjoyed for centuries. And having lived in Hawaii for many years, I can tell you that one of the major challenges to the Hawaiian population is the Western diet. And so they have had, they struggle with this because they ate a diet that was fish and very low in carbohydrate. They had fish and various fruits, but complex carbohydrates they did not have in their diet. And so now with the introduction of the Western diet, the Hawaiians struggle with obesity and type 2 diabetes more than any other culture around. I mean, more than a lot of cultures, because of the fact that historically, for centuries, they did not eat complex carbohydrates. They didn't evolve with complex carbohydrates. So it's kind of interesting. So Aiden Lawler, are you there?
I'm back here.
Yeah. So Aiden, you asked about humans and bacteria and whether or not they trigger major changes in development in our bodies or whether or not it's just minor. Well, it's really interesting to watch the development of the field because not 20 years ago, it was thought that they really only affected the tissues with which they interacted, so the gut. And then it was found that, oh my gosh, the immune system maturation requires interaction with the microbiota. And I'm talking about deep systemic immune system. And then we started to find, as we talked about in the animals in the bacterial world, that they influence the brain. So how does this happen? The bacteria are deep in the gut. How does their influence percolate to the brain? There's a really famous guy named Jeremy Nicholson at Imperial College London. And he has shown that around 18 to 20% of your metabolome, which are the small molecules in your blood, are derived from the activity of the microbiota in your gut. So what this means is that you've got microbial products. 20% of your small molecules in your blood are microbial, even though the microbes aren't there. So these microbial molecules affect each and every cell that interacts with blood. That is huge. And so every cell that interacts with blood has been over evolutionary history and is now affected by microbial products. And so that's very deep. And our whole biology is integrated with our microbes. And while we can live in germ-free environments, they have germ-free mice and, the baby in the bubble living without microbes, those people require, and those animals require very special conditions and all kinds of supplements and so on and so forth. You cannot, mammals and vertebrates, it looks like, cannot survive without, under natural conditions, they cannot survive without the microbiota. And so that's becoming fairly clear. Okay, so let's see.
We have about 5 minutes left, Margaret.
Oh, geez, okay. So,
Don't pull a Jack Gilbert. He saw how fast he could go. Don't be like Jack.
I'm trying to not. So let's see. Kimmy Ortman, are you there?
Kimmy is also someone who could not find this classroom.
Oh, she couldn't find the classroom. So she and also Jackie Penafort. Is Jackie there? Hi, Jackie.
Hi.
I talked a little bit about you asked this really interesting question about MAMPs and PRRs and what goes on with Akkermansia.
Yeah.
And Akkermansia is a microbe that has been associated in humans with leanness. And it turns out that the MAMPs, which are microbe-associated molecular patterns, which are only found in microbes, And their receptors in the host, which are the pattern recognition receptors, that's the way that animals and microbes talk. You asked about how they work, and it's all about balance and equilibria. And so Kimmy Ortman had asked this question about pathogen versus mutualist, and in fact, it's all context specific. And there's a really famous guy named Arturo Casadevall, who's the chief of medicine. He's the head of infectious diseases at Albert Einstein. And he says that there is no such thing as a pathogen or a mutualist, but it's all context specific. So E. coli in your gut is fine, but E. coli in your urogenital tract is not a good idea. And so in this MAMPS and PRR thing, it's a language that the host and the symbiont use. And what's exciting about it is the same language all the way across that spectrum from mutualism to pathogenesis, but the language is used differently. So imagine if I said to you, wow, this is on fire. And you might be sitting here with me, and what you would, by my tone, you would say, oh, she must be talking about the conversation that we're having, as opposed to, this is on fire. Everybody would get up and leave the room. So it's the way that the language is used. So it's the difference between a civil conversation and an argument, or the tone is really important. And so pathogens do something different with that language than symbionts do. Now, what Akers-Mansia does is it seems to change the mucus layer of the host and make the mucus layer more impenetrable to fats and so on and so forth, and probably to MAMPs. And so MAMPs are absolutely required. Interaction with microbe-associated molecular patterns are absolutely required for gut homeostasis, in other words, for a healthy gut. But if there's too much of them, you get huge inflammation, you get inflammatory bowel disease and all this other kind of bad stuff going on. So that's really not a good scene.
So I think we're pretty much out of time.
Oh, darn. Okay.
And I've learned an awful lot from this conversation, and I mean that sincerely.
Oh, good.
And I hope the students understand that this is, and she'll get all blushy in a minute, a great honor for you to have taken the time to speak with us, and it's true.
Oh, you're so wonderful. And listen, I just want to wish each and every one of you all the very best. I mean, you guys are getting something from Mark that is rare, and you will watch in your career, you will watch this become bigger and bigger and bigger, because it's the way that we now know the world works. It's just there are people resistant to hearing the message. So I wish you all the very best, and thank you again for this phenomenal opportunity.
Let's give it a round of applause. Thank you so much, Margaret.
Thank you.
Have a great day.
Okay, bye now.
Wasn't that bad? Okay, let me turn this off and get out of your way as quick as I can.